Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine facing a diagnosis for the most aggressive kind of brain tumor, glioblastoma multiforme.
Yeah, it's devastating.
And what's truly frustrating is how often it just, shrugs off our best treatments.
Yeah.
Surgery, radiation, chemotherapy.
It really does. It feels like we're throwing everything we have at it, and yet the tumor just persists or comes back.
Exactly. It's incredibly challenging. But what is the secret, or at least part of it, to finally making a real difference lies in understanding these tiny recycling centers within the cancer cells themselves.
That's right. What's really getting researchers excited is this idea of looking inside the tumor cells, specifically at these structures called lysosomes.
Lysosomes, okay.
Picture them as like miniature waste management facilities inside the cell. They're filled with these really powerful enzymes designed to break down cellular clutter, unwanted stuff.
So, like the cell's cleanup crew.
That's a great analogy, yeah. They're essential for a cell's everyday functions, playing a role in, everything from how cells sense nutrients to how they manage their energy.
Okay, and this deep dive is going to explore some really fascinating new research focused on how we might be able to kind of manipulate these lysosomes in glioblastoma multiforme cells. The findings suggest that by carefully looking at the balance of two fundamental building blocks, we're talking about amino acids here, lysine and arginine.
Right, two key amino acids.
That we could potentially make these stubborn tumors much more vulnerable to existing treatments, like the chemotherapy drug temozolomide.
Exactly. You shared this really thought-provoking study from Nature Communications.
Yeah, the title was Lysine-Arginine Imbalance Overcomes Therapeutic Tolerance, governed by the transcription factor E3 lysosome axis in glioblastoma. It sounds very technical.
It does, a bit of a mouthful.
But the potential impact here feels, pretty significant.
It truly does. One of the biggest hurdles in treating glioblastoma multiforme is what we call therapeutic tolerance.
Right, like resistance.
Essentially, yeah. Think of it like a weed in your garden that just keeps growing back no matter what you spray on it. Over time, cancer cells can develop resistance or tolerance to the drugs we use.
Making treatment less effective.
Precisely. This particular study takes a really innovative approach. It's searching for these fundamental weaknesses, these metabolic vulnerabilities within the tumor cells, specifically within their lysosomes. The idea is, can we exploit these weaknesses to sort of break down their defenses and make them more susceptible to chemotherapy?
Okay, so let's really dig into the core of this study then. What were the scientists actually aiming to find out?
Well, the main goal was to pinpoint these metabolic vulnerabilities within glioblastoma multiforme cells with a really specific focus on how their lysosomes function.
Okay.
They wanted to see if they could find a way to, leverage these vulnerabilities to improve how well treatments like temozolomide work and overcome that resistance problem.
Right. And what steps did they take? What kinds of experiments were involved in exploring this?
It was quite a multifaceted approach, actually. The first step involved just examining the basic activity levels of lysosomes in glioblastoma multiforme cells that they got directly from patients.
Okay, patient samples?
Yeah, and then they compared these activity levels to those found in normal neural progenitor cells.
Which are like the precursors to brain cells.
Exactly, like the baby cells that eventually develop into various brain tissues. Kind of a baseline.
And how did they actually measure this lysosomal activity? That sounds like it would be pretty intricate.
They used a pretty clever technique. Imagine you have a special dye or probe that only lights up when it's being broken down or, let's say, digested. They used a molecule that only becomes fluorescent when it's degraded by those enzymes inside the lysosomes. So the brighter the fluorescent signal they saw,
the more active the lysosomes were.
Precisely. More light meant more breakdown activity.
That's neat. So what did they find? Were the lysosomes in the glioblastoma multiforme cells acting differently than the normal ones?
Yes, they were. In the majority of the glioblastoma multiforme cell lines they tested, they showed significantly higher lysosomal activity compared to the normal neural cells.
Higher activity in the cancer cells.
Right, and here's a really key finding. They discovered a link between this higher lysosomal activity and how aggressive the tumor cells were behaving.
How so?
Well, cells with more active lysosomes showed a greater ability to form these sphere-like structures when grown in lab dishes.
And that's important because...
Because that sphere-forming ability is considered a hallmark of cancer stem cells. These are the cells thought to really drive tumor growth and recurrence after treatment.
Wow. Wow. So more active cellular recycling centers seem to be linked to the more dangerous, persistent cancer cells. That feels like a really important connection.
It is indeed. And they didn't just see it in a dish. They tested this in animal models, too.
Okay, what did they do there?
They implanted mice with glioblastoma multiforme cells that had either high or low lysosomal activity, using cells engineered to glow so they could track tumor growth. And the tumors with high lysosomal activity grew much more rapidly. And sadly, the mice that had those high-activity tumors had shorter survival times.
That really drives home how critical this lysosomal activity seems to be in the progression of the disease.
Yeah.
Now, you mentioned they were also looking at how this ties into treatment effectiveness, specifically with temozolomide. What did that part of the study show?
Right. So initially, they looked for a straightforward, direct relationship between just the overall lysosomal activity level and how sensitive the glioblastoma multiforme cells were to temozolomide.
Did they find one?
Interestingly, they didn't find a simple direct correlation.
Huh. Why not, do they think?
Well, it's likely because other factors are also playing a big role. Things like the presence of a specific DNA repair protein.
You mean 6-methylguanine DNA methyltransferase? That one's known for causing resistance, right?
Exactly that one. Its presence or absence can significantly impact how well temozolomide works, kind of independent of the lysosomes, initially at least.
Okay, so that complicated the picture. They needed to look a bit deeper.
Precisely. So they then shifted their focus a bit. They looked at another protein called CD44.
CD44.
Yeah, it's a cell surface marker that's known to be associated with resistance to therapy in glioblastoma multiforme.
Okay.
And what they observed was that the glioblastoma multiforme cells with high levels of this CD44 protein also had higher lysosomal proteolytic activity.
Uh-huh. So there's a link there. Cells known to be resistant tend to have these highly active lysosomes.
That's what it suggested. It pointed towards a potential link between how lysosomes function and drug resistance, at least in this specific subset of tumor cells marked by CD44.
Okay, so resistant cells seem to have these very active lysosomes. What did they do next to understand that connection better?
They decided to try inhibiting the lysosomes. They used a drug called bifilomycin A1.
Bifilomycin A1. And that blocks lysosome activity.
Yes, it's a known lysosomal inhibitor. They tested this drug in combination with both temozolomide and also with X-ray radiation therapy.
Okay, standard treatments.
Yeah.
And what happened when they blocked the lysosomes while also applying those treatments?
This was really interesting. While bifilomycin A1 by itself didn't really kill off many of the glioblastoma multiforme cells, it noticeably enhanced the effectiveness of both temozolomide and radiation. The cells became much more sensitive to those treatments when the lysosomes were inhibited.
Wow. Okay, that's a really significant finding then. It directly points to these lysosomes as somehow protecting the cells.
Yeah.
And maybe being a therapeutic target.
Exactly. It strongly indicated that this high lysosomal activity was somehow helping the tumor cells withstand these therapies.
So the next logical question is, what's controlling this lysosomal activity? How did they figure that out?
Good question. They then focused on a family of proteins known as the MIT/TFE transcription factors.
Transcription factors. Those control which genes get turned on or off, right?
And this particular family is known to be master regulators of how cells actually build lysosomal biogenesis. Within this family, they discovered that one specific protein called transcription factor binding to immunoglobulin heavy chain enhancer 3, let's maybe call it TFE3 for short.
Yes, please. TFE3.
Okay. TFE3 was particularly abundant in the glioblastoma multiforme cells they looked at. And interestingly, the levels of this TFE3 protein were also higher in those CD44 high cells, the ones that had the high lysosomal activity.
So TFE3 looked like a potential culprit, or at least a key player in driving this high activity. How did they confirm that?
They used genetic tools to manipulate the cells. They basically created glioblastoma multiforme cells where they turned off the TFE3 gene in knockout.
Okay.
And they created other cells where they made the gene produce more TFE3 protein than usual in overexpression.
Clever. And what did they see?
Well, just as they suspected, when TFE3 was knocked out, the lysosomal activity decreased. And conversely, when TFE3 production was ramped up, the lysosomal activity went up.
Pretty direct link.
Very direct. They also saw that TFE3 played a role in the cell's ability to form those sphere-like structures. We talked about the cancer stem cell hallmark. And importantly, it affected resistance to temozolomide.
How so?
Cells with more TFE3 were more tolerant to the drug, Knocking it out reduced tolerance.
Okay, so TFE3 seems to be pushing the activity of these lysosomes and helping the tumor cells resist treatment. You also mentioned a link to the cell's energy producers earlier, the mitochondria.
Yes, that's another important piece of the puzzle. They found that in cells with high levels of TFE3 and therefore high lysosomal activity, there was also increased activity in the mitochondria.
The powerhouses of the cell.
Exactly, the
Yeah.
ones responsible for generating energy. This suggests a pretty close interaction and maybe coordination between these two critical cellular components, the recycling centers and the power plants.
Fascinating how these different parts inside the cell seem to work together, or at least influence each other. So how does TFE3 manage to boost the activity of both lysosomes and mitochondria?
The researchers looked for genes that TFE3 controls. They identified a key target called peroxisome proliferator activator receptor gamma coactivator 1 alpha.
Whoa, another long one.
Yeah, let's call it PGC1 alpha.
TGC1 alpha, got it.
Okay, so PGC1 alpha is a known regulator of mitochondrial production, how many mitochondria a cell makes. And it's also been linked to poorer outcomes in cancer patients.
Not good.
No. So they experimented by increasing the production of PGC1 alpha in the glioblastoma multiforme cells.
And what happened?
They saw an increase in both mitochondrial and lysosomal activity. And the cells also showed some increased resistance to temozolomide just from boosting PGC1 alpha.
So PGC1 alpha impacts both.
Right. And importantly, increasing PGC1 alpha could partially undo the negative effects of turning off TFE3. It sort of rescued some of the defects.
Okay, so that really strengthens the connection. It looks like TFE3 boosts PGC1 alpha, which then ramps up the activity of both the lysosomes and the mitochondria, which ultimately helps the tumor be more aggressive and resist drugs. It's like a chain reaction inside the cell.
That's a good way to think about it, yeah.
Now, let's get to the really intriguing part, the role of lysine and arginine. How did these specific amino acids fit into this whole picture? This seemed like a key discovery.
It really was. They used metabolomics, basically analyzing all the small molecules present in the glioblastoma multiforme cells that had high versus low lysosomal activity.
And they found differences.
Yeah, they found differences in how these cells were processing amino acids. So they decided to test what happened if they deprived the cells of individual amino acids.
Took away their food source one by one.
Kind of, yeah. Removed specific amino acids from the culture medium they were growing in, and they made a really surprising discovery.
Which was?
When they specifically took away lysine, that one amino acid, it caused a significant drop in lysosomal activity.
Just removing lysine.
Lysine. And not only did activity drop, but the lysosomes themselves became more fragile, more prone to damage, something called lysosomal membrane permeabilization.
Wow. Lysine, which is an essential amino acid we get from our diet, right?
Correct.
It's actually crucial for keeping these aggressive cancer cells recycling centers working properly. That feels quite unexpected.
It is a potential vulnerability. And it gets even more interesting.
Okay.
They found that if they didn't remove lysine, but instead increased the ratio of arginine relative to lysine.
So more arginine compared to the lysine present.
Exactly. That also suppressed lysosomal activity and increased that lysosomal damage, similar to removing lysine altogether.
So it's about the balance between the two.
It seems to be, yes. The relative levels of lysine and arginine are critical for keeping the lysosomes stable and active in these cells.
And did they figure out why this balance matters so much?
Well, they found a connection to another molecule, nitric oxide.
Nitric oxide. Isn't that involved in, like, blood vessel dilation and lots of other biological processes?
Yeah.
How does it connect to lysosomes here?
Exactly. It has many roles. What they found here was that when lysine levels were low, or when arginine was in excess relative to lysine, the levels of nitric oxide increased inside the cells.
Okay.
And when they used drugs to either force nitric oxide production up or block it, they saw corresponding changes in lysosomal activity and stability.
So the idea is maybe lysine normally helps keep nitric oxide levels down, and when lysine is scarce, the extra nitric oxide messes with the lysosomes.
That seems to be the mechanism they uncovered, yes. The lysine-arginine balance influences nitric oxide, which in turn impacts lysosomal function.
That's a really fascinating interplay. So it's like lysine and arginine are competing, influencing nitric oxide, which then hits the lysosomes. And this led them to investigate something called homoarginine.
Yes, exactly. Homoarginine is a naturally occurring amino acid. It's very similar in structure to arginine, and importantly, it can also lead to the production of nitric oxide.
Ah, okay. So they thought maybe giving homoarginine could mimic the effect of low lysine or high arginine.
Precisely, and that's what they found. Treating the glioblastoma multiforme cells with homoarginine increased intracellular nitric oxide levels. It suppressed lysosomal activity, made the lysosomes leakier,
The crucial part.
and, importantly, it made the cells significantly more sensitive to the chemotherapy drug temozolomide.
Wow.
And again, just to confirm the nitric oxide link, when they blocked nitric oxide production while giving homoarginine, that increased sensitivity to temozolomide was partially reversed.
So homoarginine seems to work, at least in part, by shifting that amino acid balance indirectly, boosting nitric oxide, and thereby disrupting the lysosomes, making the chemo work better.
That appears to be the core idea, yes. It's like using a metabolite to specifically weaken the cancer cell's defenses.
Did they test this in cells that were already resistant to temozolomide, like those ones with the DNA repair protein we mentioned?
They did. And this was really promising, even in some cell lines that were resistant because they produced that O6-methylguanine DNA methyltransferase protein. Homoarginine still showed a synergistic effect with temozolomide.
Meaning the combination worked better than expected.
Yes, suggesting it could potentially help overcome this very common mechanism of resistance.
That's huge. That could be a really big deal for patients whose tumors don't respond well to the standard chemotherapy. And they didn't just look at temozolomide, did they? Did homoarginine help with other treatments?
Correct. They screened it with a range of other anti-cancer drugs and found that homoarginine enhanced the effectiveness of many of them.
Really? Like which ones?
They didn't list all of them in the summary, but they mentioned synergy with multiple other agents, suggesting a potentially broad effect. And it also increased sensitivity to X-ray radiation therapy.
Okay, so this isn't just about boosting one specific drug. This idea of targeting lysosomes by manipulating this lysine-arginine balance, maybe using something like homoarginine, could be a more general strategy.
That's certainly the hope. And what the data suggests so far, it might make tumors more vulnerable to various types of attack.
And did they take this out of the lab dish? Did they test homoarginine in living organisms?
Yes, that was the crucial next step. They conducted studies in mouse models. They implanted human glioblastoma multiforme cells into the brains of mice to create tumors.
Okay, the orthotopic model.
Right. And then they treated the mice with homoarginine, temozolomide, both or neither.
And the results?
The combination of homoarginine and temozolomide significantly slowed down tumor growth and, importantly, extended the survival time of the mice compared to just using temozolomide alone.
That's fantastic news from the animal study.
It really is. They also confirmed that giving homoarginine by itself did impair lysosomal function within the tumors in the mice, consistent with their lab findings.
This all sounds incredibly encouraging, really groundbreaking work. But of course, with any significant new research, especially at this stage, there are always limitations to consider, right?
Absolutely. It's crucial to be realistic.
What are some of the things that this study maybe doesn't fully address, or where more work is needed?
Well, the researchers themselves are quite clear about this. They point out that the precise molecular details of how disrupting lysosomal function makes temozolomide more effective still need further investigation.
Like exactly how it changes drug delivery inside the cell
Yeah.
or other downstream effects.
Exactly. For example, fully understanding how it affects the concentration or distribution of temozolomide inside the tumor cell is still being worked out. Also, remember how we talked about the close link between lysosomes and mitochondria?
Yeah, the recycling centers and the power plants.
Because they interact so closely in so many ways, it's actually quite difficult to study the effects on lysosomes in complete isolation from potential effects on mitochondria. Teasing those apart is challenging.
That makes sense. They're part of this complex, interconnected cellular machinery.
Right. And, while the results in lab-grown cells and these mouse models are genuinely very exciting, we always have to remember these are preclinical findings.
Not yet proven in people.
Exactly. Clinical trials involving human patients are absolutely necessary to confirm these results and, critically, to ensure that this approach, using something like homoarginine, is safe and actually effective for treating glioblastoma multiforme in people.
Safety first, always.
Always. And finally, they also acknowledge that the whole system controlling TFE3 and lysosomal function is likely very complex. It probably involves more factors than just the PGC1 alpha pathway they identified. There are likely other cellular communication networks, maybe involving calcium signaling or other metabolic sensors, that are also playing a role and need more research.
Okay, so fair points. More research is definitely needed, but the potential here seems undeniable. As someone who follows this field closely, what do you see as most important implications of these findings for glioblastoma multiforme research overall?
I think what's particularly exciting, really novel about this work is the approach itself focusing on the subtle but critical role of amino acid metabolism within the tumor cells.
Looking at the fuel and building blocks.
Exactly. It really highlights that we might be able to leverage these very fundamental metabolic processes, things the cell needs to survive and grow, to our advantage in cancer therapy. The concept of using a, let's call it, chemo-enhancing metabolite, like homoarginine,
Yeah, I like that term.
to boost the effectiveness of existing treatments like temozolomide is, I think, a very compelling strategy.
It feels like giving the chemotherapy a targeted boost, right at the cellular level where the resistance might be happening.
Precisely. And the potential to overcome resistance to temozolomide, even in those tumors that have that difficult DNA repair protein.
The O6-methylguanine DNA methyltransferase.
Right. That offers a potentially significant step forward for a group of patients with fewer options. Plus, the fact that homoarginine also showed synergy with other anti-cancer drugs and radiation suggests this might not be a one-trick pony. It could be a broadly applicable strategy for tackling this very challenging disease.
That's really helpful. Did they mention anything about side effects on normal cells?
That's a crucial point. The initial indications from their lab work, the in vitro studies, suggested that the combination of homoarginine and temozolomide didn't appear to significantly harm the normal neural progenitor cells,
Okay, that's good.
which does provide a hopeful sign for maybe selectively targeting the tumor cells. But again, that needs much more thorough investigation, especially in vivo and eventually in clinical trials, to understand the effects on the whole organism.
Understood. So looking ahead then, based on these findings, what do you think are the key areas for future research? Where does the field go from here?
I think there are several crucial avenues. First, digging deeper into those precise molecular mechanisms we talked about. How exactly does disrupting lysosomal function enhance these therapies? That's essential.
Knowing the why.
Knowing the why.
Hmm.
Second, identifying potentially more specific ways to target lysosomal function, perhaps finding drugs even more selective than bephyllomycin A1, or maybe refining the use of metabolic modulators like homoarginine, perhaps finding others.
More targeted tools.
Right. And third, given that close relationship between lysosomes and mitochondria, definitely more research into how these organelles interact, how that contributes to drug resistance, and crucially, how we might be able to therapeutically disrupt those interactions for benefit.
Exploring that crosstalk.
Exactly. Ultimately, the big goal here is to translate these really promising preclinical findings into effective and, importantly, safe treatments that can actually help patients battling glioblastoma multiforme.
This has been a truly illuminating deep dive into some really cutting-edge research. It really makes you stop and think, doesn't it? How these seemingly small shifts, like the balance of two amino acids in the microscopic world within our cells, can have such a profound impact on something as huge as cancer treatment.
It really does put things in perspective.
And it kind of prompts the question for our listeners, maybe. What other fundamental cellular processes these basic housekeeping tasks cells do might hold similar untapped therapeutic potential? Not just in cancer, maybe, but in other challenging diseases, too.
That's a fantastic thought to leave people with. It encourages us all to look beyond the most obvious drug targets and consider the intricate metabolic balance, the internal environment of the cell, as a potential therapeutic arena.
Absolutely. If you're interested in digging even further into the specific data and methods of this research, we definitely encourage you to look for the original article. It was published in Nature Communications.
Well worth the read if you like the details.
And as always, we'd love to hear your thoughts. Any questions you might have about this deep dive, feel free to reach out. You can usually find links to the research paper and ways to connect with us in the description or notes for this discussion.
We always appreciate the engagement.
Thanks so much for joining us for this exploration on the deep dive.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.